Good day, we are the Group 4. I am Danielle Patrice and together with my group mates, Janice Angel Costa, Rianne Hallado, and Joshua Oloya, we will discuss about the Experiment 4 which mainly tackles about the denaturation of proteins. So proteins play an integral role in the body's physiological function such as in metabolic reactions, immune response, and it also provides a source of energy. The physiological function of a protein is dependent upon its three-dimensional structure, according to Odian and Blay 2008. Hence, denaturation is important in order to unfold and uncoil the structures of a protein. Denaturation of protein occurs if the proteins are exposed to a denaturating agent such as heat, pH changes, alcohol, and salts. In this experiment, albumin is used in order to observe whether the proteins it contains will denature with the exposure of denaturating agents. Each albumin solution was exposed to the following denaturating agents, which is heat, inorganic acids, alkaloidal reagents, alcohol, and salts. The observed changes upon the application of denaturating agents were discussed and evaluated in each table in order to further understand the reactions of proteins upon the application of denaturating agents. So let us move on with the introduction. So about half of the body's dry weight is made up of proteins. Compared to other biological molecules, they carry out most of the diverse range of physiological activities. There are about thousands of different proteins inside our body, each with unique function that work together to direct and carry out the wide range of physiological processes that is necessary for life, according to Odian and Blay 2008. It was also stated in the book that is published by Odian and Blay 2008 that the physiological function of a protein is dependent on its three-dimensional structure, wherein protein structure is divided into separate levels, which are the primary, secondary, tertiary, and quaternary. One of the phenomena that causes a protein stability and structure to be disturbed is the protein denaturation. Proteins become denatured when their distinctive three-dimensional structure is subjected to alterations due to the changes in the temperature, pH, or other chemical activities which results in the unfolding of globular proteins and uncoiling of the helix structure according to BJU 2022. Usually, denaturation under mild conditions is reversible, and denaturation under extreme conditions may not be reversible. The insolubility of polypeptide molecules is often the result of denaturation due to an entangling of the chains after unraveling of secondary, tertiary, and quaternary structures according to Odian and Blay 2008. So this experiment consists of 6 glass samples filled with albumin solution that will be observed if denaturation will take place. To determine if denaturation will occur, the observers will apply heat and add inorganic acids, alkaline reagents, alcohol, and salt to the albumin solution to observe whether denaturation will occur to each glass sample filled with albumin solution. This experiment intends to determine the process of denaturation of proteins, learn what factors affect protein denaturation, and identify whether all proteins denature at the same temperature. Through this, it will also help us understand why egg whites and milks are used as antidotes for heavy metal poisoning. So that is all for my part. I am Danielle Patrice. Thank you.
Good day, everyone. I'm Janice Angel Costa, and today I will be discussing the methodology of experiment 4 entitled The Naturation of Protein. So, the protein contained in egg whites or albumin was subjected to several denaturing methods in the effects of heat, pH changes, alcohol, and salt. So, the solution is made from egg whites and water will be used to prepare the albumin water mixture. A small glass jar, spoon, and transparent container are the tools used. To begin the experiment, separate the egg whites and yolks from two eggs. Then water should be added to the egg whites in a clear glass of container. And the mixture should be stirred well. So first, in the effect of heat, the solution that is used is the albumin water mixture, and the apparatus that should be used are small glass jars, tablespoon, masking tape, and a thermos with hot water. So the procedure is first to place 5 ml albumin solution into separate glass jars. Using the masking tape, we can label 1 and 2. So in number 1, add 3 ml distilled water and 10 ml hot water, then stir. And in number and add 3 ml of distilled water again in jar 2, then stir and observe and compare the result with jar 1. So second, in the effect of pH changes, the solution that is used is an albumin solution and the chemicals used are calamansi juice and baking soda. So the apparatus that is used are tablespoon, a transparent container, and masking tape. Using a masking tape, label each container with 3 and 4. Then put 5 ml albumin solution in container 3. Then add 2 ml of calamansi juice and add a pinch of baking soda in jar 4 with 5 ml albumin solution. Then observe and record changes and compare differences or its similarities. The third one is in the effects of alcohol. So the solution that is used is an albumin solution again, while the chemical used is ethyl alcohol. So the apparatus that is used in this procedure are tablespoon, transparent jar, teaspoon, and the masking tape. So the procedure is to first place 5 ml albumin solution and using the masking tape, label it 5, then add 3 ml of 70% ethyl alcohol. After adding that, observe and record the changes. Lastly is the effect of salt. So the solution that is used is an albumin solution and the chemical used is salt. So the apparatus that is used are tablespoon, transparent jar, and masking tape. The procedure is to first add a pinch of salt with albumin solution in a jar and label it 6. Then wait until the reaction occurs. After that, observe and record the changes.
Good day, I'm Joshua Oloya, and together with Ms. Rianne Hallado, we're going to discuss to you about the discussion of the previous experiment, which is the experiment 4, entitled The Denaturation of Proteins. So first, let us talk about proteins. As said earlier, proteins are complex molecules that made up of long chains of amino acid that defines their functions. Now, when a protein is denatured, it loses its three-dimensional shape and as a result to its biological activity. Now, some of the factors that might cause the denaturation of proteins are the changes in temperature or pH level or potential hydrogen or exposure to certain chemicals or radiations. Now, these factors can disrupt the bonds and interaction that hold the protein in its functional shape, causing it to straighten out and lose its specific structure. So now to discuss it more further, here is Ms. Rianne Hallado to discuss to you the process of denaturation of protein.
As an addition, protein has three dimensions which are primary, secondary, and tertiary. Primary, it is the sequence of chain in amino acids. Secondary, it is the sequence of amino acid are linked by the hydrogen bonds. Tertiary occurs when certain attractions are present between alpha helices and pleated sheets. However, it has also a structure of what we call quaternary. Quaternary is protein structure that consists of more than one amino acid chain that assembles to form a larger biologically active protein complex. Overall, the structure of a protein determines its function as it governs the interaction of the protein with other molecules in its environment.
Thank you, Ms. Rianne. Now, going back, as I said earlier, is one of the factors that may affect the denaturation of proteins is changes in temperature, specifically heat. So how does heat affect the denaturation of protein? Now, it affects the denaturation of protein by producing a coagulations. How? By making molecules vibrate too violently. Now, as you can see in the table 1, which is a heating, the samples are albumin and gelatin with and without hot water. So theoretically speaking, the sample albumin with hot water supposedly to have a violet in color and observation and should be negative in the denaturation because it has a presence of protein and it should be react with the heat, meaning there was an error that occurred in the previous experiment. So one of the factors was mainly because of the time duration that the albumin was stuck in the container or it did not stir properly or rapidly. So as for the other solution, it turns out that they are negative in the burette test and positive in the denaturations as it should be. Now, for inorganic acid, as I said also earlier, one of the factors that may cause the denaturation of proteins is changes also in pH level. So, how it disrupts hydrogen bonds and salt bridges. Now, all of the albumin sample solutions in table 2 coagulate as shown also in the table 2. So indicating that they are positive for denaturation, and the main reason is because is that protein makes up albumin. And when a protein is exposed to an inorganic acid, it destroys the hydrogen bonds and salt bridges. Now, this causes the prolonged action result in the real hydrolysis of peptide bonds. Now, the sample solution for gelatin is protein-free, meaning it is a lack of protein to gelatin. So nothing happens when you expose it to inorganic acids.
Now that we are done with the first two components of our experiment, we will now proceed in discussing the alkaloidal reagents. Alkaloidal reagents are naturally occurring compounds with diverse biological activities. It is commonly used to disrupt the protein structure by breaking the non-covalent interaction that hold the protein together, such as hydrogen bonds and hydrophobic interaction. So with the result that we gathered during the experiment, components of albumin with sodium bicarbonate and sodium hydroxide coagulate and became positive with denaturation. However, it goes opposite with gelatin, because its hydrogen bonds are relatively stable and resistant to disruption by alkaline reagents. Next, we have the table for alcohol. For its result, we have a little confusion, particularly with its observation. But first, I will define alcohol. Alcohol refers to a group of organic compounds that contain a hydroxyl group, which is attached to a carbon atom. The most common type of alcohol is ethanol. Alcohol can act as a denaturant by disrupting the weak hydrogen bonds that hold protein together, causing them to unfold and lose their three-dimensional structure. But going back with the result, both albumin and gelatin coagulate with alcohol, since alcohol can cause denaturation of protein that disrupts the weak chemical bonds that hold the protein structure together. However, even if gelatin coagulates with alcohol, it is still negative with denaturation, since as we all know, gelatin has a natural composition of solidifying. It does not denature because the protein chains are already disordered and flexible state. Instead, the alcohol can interact with water molecules in gelatin solution, leading to changes in the texture and other properties. Lastly, for the salts, the most common effect of salt on protein is to induce protein denaturation or unfolding. This is because salt ion can disrupt the electrostatic interaction between charged group on the protein surface, destabilizing the protein structure and leading to denaturation. Moreover, with the results that we have, high salt concentration causes the protein to precipitate or decrease the solubility. Since salt ions, in this case, compete with the protein molecules in binding water molecules. This interaction tends the protein to coagulate in the mixture that results in a positive denaturation.
Next is the conclusion and recommendation of experiment 4. So, first is the conclusion. A protein structure can be altered by a number of conditions known as denaturing agents, including temperature, pH, salinity, and solvent polarity. Any one or a combination of these parameters that deviates from normal will cause the protein structure to change. This change is often referred to as being denatured. The egg white or albumin proteins are denatured in the experiment when hot water, calamansi juice, and alcohol are added to each container, causing the egg white to change color from clear to white in all of the distinct ways. Because hot water, calamansi juice, and alcohol are the denaturing agents that are needed to break down a connection in protein denaturation. This study demonstrated how egg white proteins, as well as the texture and appearance, can be altered by the denaturing chemicals. As for the gelatin, it does not coagulate or denature based on the experiment, so we therefore conclude in this experiment that the rate of denaturation, conveniently followed by the amount of insoluble protein obtained on neutralization, varies with different proteins. This may serve for the differentiation of proteins. Next is the recommendation. So, denaturation with the use of cold temperature was not implemented in the experiment. Thus, it is recommended to execute the experiment with the use of lower temperature. On top of that, when conducting experiment involving proteins, it is important to take steps to ensure accurate and reliable results. Reliable result. One of steps that we must consider is to minimizing or exposure to heat during experiment involving protein samples. It is important to minimize exposure to heat as much as possible. Samples should be kept on ice or at low temperature when not in use. Additionally, if heat is required for the experiment, it should be kept as low as and for the shortest duration possible.
And that is all for the experiment number 4, which is the denaturation of proteins. So once again, we are the group 4, and thank you for listening to our simple discussion. Thank you.